Welcome to Pathology Central Key Concepts. The topic of this video is mutation mechanisms. So in Chapter 4 of Robbins and Kumar Basic Pathology, we focus on the different mechanisms of mutation and associate them with diseases that manifest from these different mutations. But I think it's useful, instead of focusing on all the different diseases, to have a video where we just talk about the mutation mechanisms, so you can have a solid foundation in this, because you'll see these again and again as you work your way through Robbins. So with that in mind, I'm going to discuss chromosomal translocations, non-disjunction, trinucleotide repeat amplification, and genetic imprinting, with a quick flyby of mitochondrial mutations. Let's begin with chromosomal rearrangements. These are perhaps the most straightforward of the types of mechanisms I'll be discussing. In chromosomal rearrangements, you have chromosomal breakage and subsequent loss or rearrangement of material. Now, of course, chromosomes can break and rejoin. That will not lead to a rearrangement. There are multiple different types of chromosomal rearrangements. I've listed them here. I'm going to describe them here, and then I will show you a nice figure from Robbins and Kumar Basic Pathology that makes it a lot easier to visualize. You can also turn to that figure in your book. So chromosomal translocations occur when we get the transfer of a part of one chromosome to another. Now, the two types of chromosomal translocation that are relevant for human disease are reciprocal balanced translocations and centric fusion, also known as Robertsonian translocations. And the reason these are important is because it's possible to be viable with these types of chromosomal translocations. For example, unbalanced translocations are typically catastrophic to the cell. So in a reciprocal balanced translocation, we have no loss of genetic material, and the manifestation of this will depend on when the translocation occurs. So for example, if it occurs in a gamete, then the offspring will have all of their cells with this reciprocal translocation. Because there has been no loss of genetic material, depending on where the chromosomal translocation occurs, they may be perfectly healthy and show no phenotypic difference at all. However, since this reciprocal translocation is throughout all of their cells, it includes their gametes, and they will have unbalanced gametes that can have an effect on their own offspring. Also, if you get a reciprocal balanced translocation in the somatic tissues, this can give rise to malignancies such as synovial sarcoma. Now, centric fusion or Robertsonian chromosomal translocations occur with your acrocentric chromosomes. Those are the ones with a very short, minimal P arm and a longer Q arm. And what happens here is we're going to get those fusing together with loss of our short arm material. And again, although the individual who is the product of this sort of fusion from their parents' gametes may have a perfectly healthy phenotype, they will again have unbalanced gametes that can affect their own offspring. In isochromosomal rearrangements, we have a division of the centromere horizontally instead of vertically, typically leading to loss of one arm. Deletions are quite straightforward, simple loss of a fragment. In inversion, we have those two breaks, and then that piece of DNA flips, and we get reuniting. And then finally, we have ring chromosomes, which are a variant of deletions, where you get breakage at the tips of your chromosomes, and then they reunite, forming a ring. So this is the figure from Robbins and Kumar Basic Pathology. I'm going to go through each of these, beginning with our translocations. So here we have our balanced reciprocal translocation, a break in two separate chromosomes that then swap material. A classic example of this would be synovial sarcoma, characterized by a balanced translocation between the X chromosome and chromosome 18. The centric fusion or Robertsonian translocations occur, as I mentioned, in acrocentric chromosomes. You can see them listed here. And this is relevant for human disease. For example, if we get a centric fusion of chromosome 21, we're going to lose this material from the short arm, but this will have two copies of the long arm of chromosome 21. And then when, if this is in a gamete with the second wild type chromosome 21 from the other parent, that will lead to trisomy 21 and Down syndrome. Now, this is an uncommon mechanism by which Down syndrome arises, but worth being aware of. We can also get our isochromosomes, where we have this breakage through here horizontally instead of vertically, with a typical loss of the short arm. And we see this in about 15% of individuals with Turner syndrome. Now, I don't think it's important for medical students to memorize these exact percentages, but it is important to understand that there are different pathways that can lead to the same syndrome. And this accounts for some of the variability that we see in the phenotype. The disease that's associated with deletions in this chapter is 22q11.2 deletion syndrome. Quite simple, 2 breaks, loss of material leading to this syndrome. And then another type of deletion, as I mentioned, is our ring chromosomes, where we have breaks here at the ends of the chromosomes that then circularize with loss of this material. This is something that we see, for example, in well-differentiated liposarcomas. And then coming back here to inversions, we can either get breaks that occur on one side of the centromere or ones that frame the centromere, leading to paracentric and pericentric inversions respectively. And then just to associate a disease state with this, we know that we can see inversions in about 43% of patients with hemophilia A. So chromosomal rearrangements, I think, are pretty straightforward. Let's talk about something where it gets a little bit messier. That is non-disjunction. So what happens here is you have a failure during cell division. So for example, you can get failure of a homologous pair of chromosomes to separate at meiosis 1, failure of sister chromatids to separate at meiosis 2, or failure of sister chromatids to separate in mitosis. So let's work through each of these individually, beginning first with a discussion of normal meiosis. So this is just an example with a parent cell with a 2n equals 2. In meiosis, we're going to get duplication of our genetic material. And then as we go through meiosis 1, we're going to get cell division with one chromosome going to each daughter cell. Then to form gametes, we will have cell division again without replication, yielding n equals 1. So we have our 2n to our n cells. Now, what we see in non-disjunction in meiosis 1 is a failure of the homologous pair of chromosomes to separate. So here, instead of each of these going into a daughter cell, we have 2 going into one daughter cell and none to this one. This is going to lead to trisomy in 2 of the gametes and monosomy in the other 2. The reason this math looks a little complicated is because keep in mind that 2n in humans is 46. And this would be a very complicated figure, so just understand that there are a whole bunch more chromosomes in here. Now we can see this particular meiotic non-disjunction in meiosis 1. It's the most common cause of Down syndrome, so 75% of children with Down syndrome. We also can see it in Klinefelter syndrome and Turner syndrome. Now we can also get meiotic non-disjunction in meiosis 2, and in this we get failure of the sister chromatids to separate at meiosis 2. So here we have our correct homologous pair of chromosomes have separated at meiosis 1, but here the chromatids, instead of separating appropriately, are going to lead to one daughter cell with a trisomy, one with a monosomy, and then we have 2 that have the appropriate number of chromatids. And then finally, we can get mitotic non-disjunction, and what this will cause is mosaicism. And what we see in this is failure of the sister chromatids to separate in mitosis. So we can see here that we end up with a trisomy in one daughter cell and monosomy in another. And it's important to recognize that depending on when this happens, you can get variability in the phenotype of these different syndromes. So Turner, Down, and Klinefelter syndrome can be due to mosaicism. So let's look at what the consequences of mosaicism look like. This is a really nice figure that I think illustrates it well. So you have the wild type sperm and egg yielding a zygote that then undergoes mitosis. And at some point here we have it in the 4-cell stage. There's some sort of genetic aberration. And all of the progeny of that cell will have that same genetic change. And they're going to form these various populations in the developing embryo. And if you were able to mark this in some way, you could see there would be patches of populations in the adult human. Now, this is not typically what we see in people, but there's a nice illustration that shows these different populations here in a tortoiseshell cat. So this is a female cat with patches of orange and black. In cats, the gene for coat color is on the X chromosome, and we get X inactivation, which occurs randomly. So this population of orange fur is going to have developed from one cell that had X inactivation that led to the expression of this orange allele, whereas this black area is going to have the opposite. So I just think it's a nice example. So let's now return to humans with trinucleotide repeats. This is an interesting mechanism of disease. What happens is we have long repeating sequences of triplet repeats. And as you know, during DNA replication, you can get inappropriate expansion or even deletion of these nucleotides. What we see in these trinucleotide repeats is as we increase in our number of repeats, this is going to correlate with whether the individual will be healthy, have what we call a premutation or a mutation. Now, these expansions can occur anywhere associated with a gene, so it may be in the promoter, in the 5' or 3' untranslated region, in an intron or an exon. And depending on where these expansions occur and in that particular gene, what the effect is, we can see a couple of different manifestations. So one is these expansions may completely suppress protein synthesis, for example, something in the promoter region. And this will lead to a loss of function. We can also get expansions that are toxic because we have a long string of amino acids leading to misfolded proteins. This can cause the misfolded protein response and cell injury. Now, a classic finding in the trinucleotide repeat diseases is something called anticipation, which is that the clinical features worsen with successive generations. And this is because, again, we get expansion during oogenesis or spermatogenesis, which is going to correlate with health, premutation, and mutation. So let's take a look at a figure that just gives some examples of diseases and where these expansions occur. Now, I'm going to go into a significant amount of detail on the Fragile X syndromes here. We also have a very straightforward triplet repeat that I think is good to illustrate the disease process. So in the gene that encodes the protein Huntingtin, we get in this exon a CAG triplet, which results in long strings of glutamines. And these long strings are going to prevent accurate folding. This leads to a toxic gain of function and causes cell injury. Now, I say this is a fairly straightforward disease because we know that less than 35 copies, healthy, greater than 35 copies, we have pathogenesis. And the reason this is, I say, more straightforward is that when we talk about here, it's really in Fragile X that we talk about mutations, premutations, and health. So here it's just either healthy or mutation. And in Huntington disease, with increasing copy number, we get decreased age of onset. So this is that concept of anticipation. Expansion occurs during spermatogenesis, so it is only with paternal transmission we'll see anticipation. So if the mother has Huntington disease, her offspring will not have a further expansion of those CAG triplets. Okay, let's now approach Fragile X syndrome, which is listed here as really 3 separate sorts of diseases, or 3 separate syndromes. So why is it called Fragile X? Is it that the chromosome breaks? No, but it looks broken. So here you can see the X chromosome, which has been stained with typical banding dyes that we use in classical cytogenetics. This is a map of the X chromosome, and you can see that we have this expansion. This is where we get the expansion in Fragile X syndrome, and you can get up to 4,000 copies of this particular triplet, and that area does not stain well with our dyes, and so it looks like we're missing a chunk. So it looks like there's a tip of it is floating off here, so it looks fragile. So what is this area? This is a gene called FMR1, and the product is called FMRP, or familial mental retardation protein. So this protein is widely distributed in the body, but is in increased levels in the brain and testis, and that's relevant to the symptoms that we see. What this protein does is that it is going to enter the nucleus. It binds and transports specific mRNAs to our axons and our dendrites. And what it does there is it's going to regulate mRNA translation at our synaptic junctions. And this is relevant because our synapses are very important for learning and memory. And what is particularly interesting and complicating about Fragile X is that this single gene is responsible for what appears to be 3 syndromes. So let's take a look at that. Okay, so this is Fragile X syndrome, also known as Fragile X Tremor-Ataxia, and then also Fragile X premature ovarian failure. So 3 separate syndromes. So we first learned about Fragile X syndrome, which is characterized by intellectual disability and macroorchidism, which is manifested as larger testes, because as an X-linked disease, these manifestations were seen in boys. Now, we learned that with Fragile X syndrome, this was due to expanded repeats in the 5' untranslated region, and expansion occurs during oogenesis. Now, as we learned about this disease, we saw that 6 to 52 repeats was a healthy individual. The average is typically about 29. And we knew that more than 200 repeats, we had actual Fragile X syndrome with intellectual disability and macroorchidism. What we thought happened is in between these 2 limits, we had what we called a premutation. And it was thought that there really wasn't a lot going on in individuals who had a premutation. But what we've actually learned is that individuals with a premutation do actually have a phenotype. So what happens is that in this premutation, this causes a toxic gain of function. And this expanded mRNA, which has these increased repeats in the FMR1 mRNA, will sequester mRNA binding proteins, leading to intranuclear aggregates. So we have a toxic gain of function with cell injury. What we see in more than 200 repeats is what we call the full mutation. We get methylation, and none of this protein is formed at all. So what we see here is that in the male premutation, about 50% of individuals with this number of repeats will develop a progressive neurodegenerative syndrome in the 6th decade. Now, it took us a while to figure out there was a link between Fragile X syndrome and this Fragile X Tremor-Ataxia, because the neurologists who took care of the pediatric population weren't the same as the ones taking care of the elderly population. Now, if we look at what happens with the male mutation, so greater than 200 repeats, full mutation, no protein, about 96% of these boys will have developmental delay and or intellectual disability, and about 90% will show macroorchidism. Now, we also have something unusual for an X-linked disease in that females are also affected. Typically, we think we have a healthy wild-type X chromosome, so we'll be fine. But actually what we see is that in the female premutation, about 20% of individuals have mild cognitive impairment as well as this premature ovarian failure. And when we move on to the full mutation, about 30 to 50% will have mild cognitive impairment, and about 20% will have early reproductive failure. So let's take a look at this in a kindred. You can see here we have a couple of different examples. So we have our carrier male. He's got an X chromosome here. You can see this little red area is the expanded area, so less than 200 repeats. Here is his wild type Y chromosome, and here's our wild type female with 2 wild type X chromosomes. So he has this premutation, so about half of individuals with this genotype will have neurodegenerative disease in the 6th decade. Now, looking at their offspring, we have one son who has the wild type X and a healthy phenotype. We have a second son who also is wild type and normal phenotype. And then we have 2 carrier females. So in this carrier female, she has the premutation that she inherited from her father. Remember, this expands during oogenesis. And so she is either going to be healthy or she may be affected, because remember, about 20% will have this mild cognitive impairment or premature ovarian failure. Now, if we look at at her sister who has offspring with a wild type male, you can see that this is going to expand during oogenesis, and so 2 of her offspring are going to receive the full mutation. Her son is going to be affected. He's going to show that intellectual disability, developmental delay, macroorchidism. And her daughter is going to have 1 wild type chromosome and one full mutation chromosome. And about half of these will be affected, often mildly. All right, so I think that this is a very complicated disease to talk about triplet expansions, but I do think that Huntington disease is a good way to frame this concept for yourself. So this brings us to the last of the really complicated mutation mechanisms I want to discuss, which is diseases of imprinted regions. Now, you probably learned in high school biology, you get chromosomes from your mother and chromosomes from your father, and it really doesn't matter, they're all the same. Not true. Now you're in medical school, and we know that there are functional differences between maternal and paternal copies of some genes. And this is due to a process called genomic imprinting, in which we get epigenetic inactivation during gametogenesis. And this can occur through promoter methylation or modification of DNA binding histone proteins. Now, the terminology is that maternal imprinting refers to transcriptional silencing of the maternal allele. Paternal imprinting refers to transcriptional silencing of the paternal allele. Now, how do we even learn about this? Because even if we look at them on classical cytogenetics, the chromosomes look identical. You can't really tell by looking at them which comes from the mother, which from the father. And the way we learned about this was that we have 2 syndromes that seemed completely unrelated. Prader-Willi syndrome and Angelman syndrome. Now, while both have intellectual disability, the former also has these additional findings: short stature, hypotonia, obesity, etc. Whereas in Angelman syndrome, we had ataxia, seizures, and inappropriate laughter. So 2 apparently completely different diseases. But using classical cytogenetics, it was found that about 60 to 75% of these kids had the same chromosomal abnormality, deletion in chromosome 15 from band Q11 to Q13. So how does this happen? So this occurs through imprinting. So this is a figure from Robbins and Kumar Basic Pathology. You can see here, this is chromosome 15. We have our maternal chromosome and our paternal chromosome. And in this area, we have these 2 sets of genes. We have our Prader-Willi genes and our Angelman genes. And in the maternal chromosome, the Prader-Willi genes are turned off with active Angelman genes, and the converse is true in the paternal chromosome. So if you get a deletion in this area in the maternal chromosome, you're going to lose your active Angelman genes, so the patient will have Angelman syndrome. However, if we lose this area through deletion in the paternal chromosome, we now are going to lose our active Prader-Willi genes, and the patient will have Prader-Willi syndrome. So this seems pretty straightforward, I think, when you think about it and you see this figure, but it can actually get even slightly more complicated. And that occurs because we found that that there were some children with Prader-Willi and Angelman syndrome who had 2 completely normal chromosome 15, so no deletion at all. How did this happen? It happens because both copies came from either the mother or the father because of something called uniparental disomy. And what happens is if both chromosomes come from the mother, then we have lost our active Prader-Willi genes. Conversely, with the father, we will end up with Angelman syndrome. So now for our quick flyby, just something that I think is pretty interesting is mutations in mitochondrial genes. Now, as you've all learned, mitochondrial genes show maternal inheritance. This is because the egg is much, much larger than the sperm, so almost all of our mitochondria come from our mothers. Now, mitochondrial DNA is going to encode enzymes for oxidative phosphorylation because that's what the mitochondrion does. And it's going to primarily affect tissues that do a lot of oxidative phosphorylation, such as these. And it's going to show, as I mentioned, this maternal inheritance. This is a kindred that you might see, for example, on a step exam. So presumably this brother and sister, their mother had a similar disease. She passed it on to her son, but because even though his mitochondria are affected, none of his children will be affected because he's only passing on a couple of mitochondria with his sperm. His sister, however, is going to pass this on to all of her children. It will end again with her son, but both of her daughters will pass it on to all of their children. All right, as always, here are some questions. Feel free to pause and see if you can answer them. If not, maybe watch the video again. As always, thank you for your time and attention. I hope you found this useful. Please follow me on Twitter, and I really appreciate comments down below. Thank you.